Hi everyone, I'm going to talk about a project I've been working for a few years now, and it's about the conservation of one of the most endangered reptiles in Europe. The species Testudo hermanni is a tortoise. There are two subspecies in Europe, one in the Western Europe, the other one in the Eastern regions. This species is protected and is classified as near threatened by IUCN Red List. But if we look at the Western subspecies, we see that this was classified as endangered in 2004. So why this species endangered? Because the habitat reduction, especially in Western countries, also because the forest fires that increased in the last years, and also because the intensive harvesting for pet trade. And especially for this reason here, in the last 50 years, there have been around 1,000 of tortoises released illegally, principally from Balkans to Italy and other Western countries. So in Italy, especially, we have many rescue centers with tortoises of unknown origin and difficult to manage. This is a big issue, but it can be also a potential resource for introduction plans in the future. But to do this is crucial, firstly, to understand the effective risk of translocation and hybridization in the species, and then propose management plans for the conservation based on genetics and genomics analysis. So in the previous studies, we identified the genetic structure in two subspecies. We analyzed a large dataset across Europe. We genotyped the samples at 7 microsatellite loci. We identified 6 genetic clusters, 2 in the Western subspecies, in the Eastern subspecies, and 4 in the Western subspecies. So then we selected a subset of samples. We performed ddRADseq sequencing. We obtained a few thousand SNPs, and we confirmed the previous results, but also we are able to see a substructure, especially in this region here in south of Italy, where we identified 3 genetic clusters that we didn't see before. So after these 2 studies, recently we decided to generate high-quality genome for this species in order to improve our knowledge in conservation, also in the genomics and evolution of this species. So we sequenced the genome from a captive tortoise from a zoo in north of Italy, Parco Natura Viva. We sequenced PacBio, HiFi, and Nanopore from these samples. This is a female. And we also estimated the genome size of the species, and we found that it was 2.1 gigabase approximately, similar to other turtle species. And also we estimated the heterozygosity, and we see that it's 0.3% of the genome. So we assembled the genome. It's high quality genome. We see that we have only a few low number of contigs in our assembly. Also, we see that it's high complete. We recovered 97% of BUSCO. And also we see that gene model we generated from the genome assembly is comparable with the BUSCO score that we got from the genome assembly. So the first thing we did was to do a phylogeny with our assembly. We used also other genomes of turtles available, and we made this phylogeny. We see that our species, it's this clade here, is a family called Testudinidae. There are not so many genomes available in this group of turtles, so we believe that this assembly can be important resource not only for population genomic analysis of this species, but also for comparative genomics of this group of reptiles. So the next step was to investigate the population genomic for this species. So we resequenced the whole genome of a few tortoises from different genetic clusters that we identified before with different markers. And we see here, we sequenced the genome from Eastern subspecies, from Greece population, from Northeast Italy, and also from the samples here that we use for the genome assembly. And also we sequenced the genome from the Western populations, and we sequenced the genome from Tuscany in Italy, also from Sicily, and also from a little population in France. So the first results, the preliminary results, we confirm a clear distinction among these 2 subspecies. We see from this PCA, we got the 2 subspecies in blue and red. We see that there is high divergence between these 2 subspecies. Also, we see the first component explain almost 90% of the variance. And also we see that there are a substructure within the subspecies. This is confirmed by the admission analysis. We see that we identify different clusters And we see also that especially in Western population, Western subspecies seems to be more admixed with respect to the Eastern populations, at least for the population that we are analyzing here. So the next step was also to focus more on the inbreeding level and also genomic diversity, investigate these patterns in our populations. So we first analyzed the proportion of the genome in long row, and we see from the plot that we got different results, variable results across the population. So we got very low percentage of the genome composed of row of random homozygosity in the Greece population, for example, but we got a very high percentage, like for almost 40% of the genome composed of row in the France population. And also we estimate the genomic diversity in this population, and we see here also that it's quite variable across the samples, across the populations, and also here again the France population seems to have a very low genomic diversity, and this can be compared to other reptiles that are considered vulnerable or critically endangered. So we expected this because the France population is very isolated and it's a very small population. So we believe that these results are still preliminary, but can be very important in the future to evaluate better the conservation status of these species. So in the next step, we want to investigate the demographic history of these species and the populations of these species. We also want to estimate the divergence time between the two subspecies. And also we want to analyze the genes that are potentially under selection, and we want to identify if there are genomic signature in the Western and the Eastern subspecies. And we believe that all these analyses, with these other analyses, these results, we will get to the main goal that I mentioned at the beginning of the presentation, that is to understand the effective risk of translocation, hybridization in species, and then help the management plans for the conservation of the species in the future. So I want to thank all my colleagues working on this project, and also I want to thank you for listening. Thanks.